Kim Orth, Science.
So now you're going to try to do it without centrifuging and just see what happens when they infect.
Yeah.
Dr. Kim Orth's interest in bacteria is infectious. She and her students at UT Southwestern Medical Center in Dallas spend countless hours studying the germ.
It excites me that we can discover new things by studying bacteria because they figured it out so long ago.
What they figured out was how to manipulate the signaling system in our cells, the system that triggers our disease-fighting defenses.
They basically hit the Achilles heel in a signaling pathway, and they can shut the whole pathway down.
Kim believes if we can learn to manipulate cells the way bacteria do, the possibilities could be enormous. A native of Plano, Texas, Kim had no real interest in biochemistry until college. She took a class at Texas A&M on molecular genetics, and the professor ignited a spark.
I'd always learned biology and chemistry and genetics. And he took them all and he melded them all together, and I thought, this is amazing.
Kim earned her bachelor's degree in biochemistry, then her master's from UCLA and her Ph.D. from UT Southwestern. Kim was working on her postdoc at the University of Michigan when she made an amazing discovery. She found that the bacteria that cause bubonic plague spread by sending what she describes as targeted missiles into cells. Those so-called missiles prevent proteins in the cells from sending out signals for help. Kim compares it to taking control of the light switches in a house.
So the bacteria turns off all the switches so that the cell can't change shape and can't engulf the bacteria, right? And so we figure out what those switches are, and in doing so, we figured out new switches.
Those new switches represent previously unknown ways in which our cells work. Impressed by her discovery, administrators at UT Southwestern asked Kim to open a lab on their Dallas campus to continue focusing on her promising research.
She's laying the groundwork for our understanding of how bacteria cause disease. That understanding is essential for the eventual development of therapeutics, treatment of a number of diseases caused by bacterial infections.
They shouldn't be able to bind because they'll take off the fibronectin.
The breakthroughs have continued. For instance, Kim and her researchers have discovered how bacteria in shellfish destroy cells in the human body.
I think Kim's work is pioneering and creative. She's used a multidisciplinary approach that's included biochemistry, cell biology, structural biology, and infectious disease. And she has opened up a whole new area of our understanding of bacterial pathogenesis. And her work has brought enormous attention to our institution.
Kim believes she couldn't be working in a better place.
There's so many experts on so many parts of the cell that you can have wonderful collaborations with many different scientists. What's really important right now is that we keep investing in basic research, because if we don't, then we won't be able to make the tools for medicine later. I feel so lucky that I've been supported by people in order to do this basic research.